For those of you who've come last year, you might remember Sam, who talked about plumbing to us. It's a very memorable talk. Now, Sam Bryen is actually the MD New South Wales PhD Scholar, having done a Bachelor of Science degree in zoology before. She's now doing research into genetic variants which impact splicing, which then translates into clinical care for patients and their family, and she's working in the Children's Hospital Westmead. So we'll pass the time to her now to talk about taking control of splicing, new therapies for neuromuscular disorders.
Hi everyone. So my group at Westmead Children's Hospital, we focus on trying to find the genetic answers for patients with neuromuscular disorders. This can be quite challenging at times because each individual and every family has their own set of unique variants to them, thousands of them in fact. So it can be very difficult to sift through these and try to figure out what is the cause of neuromuscular disorders. So we have a research cohort of roughly 200 families. And what we found, we have diagnosed about 60% of these, but what we have found is that about 1/3 of these families have at least one splicing variant. So my PhD focuses all about the process of splicing and variants that affect the process of splicing. And I talked about that last year. So this year I'm going to focus more on what you can actually do once you have a genetic diagnosis. So it can help guide clinical management. And also allow for family planning and prenatal genetic diagnosis. Also, in addition, it can sometimes pinpoint certain treatment methods that are available and also allow for eligibility for certain clinical trials. So today I wanted to talk about Antisense Oligonucleotide Therapies. It's a big name. There are 2 antisense therapies that are currently approved by the FDA over in the States for neuromuscular disorders. And today I wanted to talk about eteplirsen as an example, which is used to treat some cases of Duchenne's muscular dystrophy. So I had a look on Wikipedia as to see what it said about eteplirsen, and it says, eteplirsen is a morpholino antisense oligomer which triggers the excision of exon 51 during pre-mRNA splicing of the dystrophin RNA transcript, which might not mean much to many people. So I was hoping to try and break down this sentence today to sort of help people understand what this technology is about and what it can do for people. So first step, what is pre-mRNA splicing? So to answer that question, we need to start from the beginning. DNA is the set of coded instructions that make you who you are. So very much like zeros and ones in the computer world, we have A, G, C's and T's in our cells to tell our cell what to do. This is sort of arrayed in a linear sort of sense, and you can see the A, G, C's and T's sort of strung on a backbone, and these will always pair with another single strand, and together each little pair, we refer to them as base pairs. So you see that G's will always pair with C's and T's with A's. And this forms this double helix, which you might have seen before. So we actually have 3 billion base pairs of information in every single cell in our body. And these are organized into 23 pairs of chromosomes, as seen here. So just imagine that double helix shape wound up even tighter to sort of make these structures that you can see here. So genes are the instructions that make proteins, and they're scattered throughout these 23 pairs of chromosomes. And there are 20,000 genes that we know of. And over 600 genes have been associated with neuromuscular disorders. So scattered throughout these 600 genes could be the answer that is the reason why neuromuscular disorders occurs, and finding it can be difficult. So I'm just going to give 2 examples today. So the Dmd gene and the Emd gene, which you can see pointed out in this X chromosome here. So the reason I've chosen Emd is it's actually quite a small gene. You can see it's 2,000 base pairs long. So this giant blue box represents A, G, C's and T's, and that's all you need to make the protein Emd. But what actually happens is we don't need all of that. We only need 700 base pairs. So these exons, these blue boxes, are the coding parts. So we only need those coding parts to make the protein. These non-coding intronic sections that I've shown with this blue line actually need to be removed, and we ignore this information, which is very interesting. The Dmd gene is, by comparison, absolutely massive. It's the biggest gene. It spans the biggest length in our genome, with being 2 million base pairs long. And we need comparatively only a very small amount of this information to actually make our dystrophin protein. So we need 11,000 of these base pairs. But something in our cell has to sort of stitch this information together and get rid of the stuff that we don't really want. Only 2% of the entire genome of that 3 billion base pairs is actually used to make protein at the end of the day. So how this works is we have our DNA in every single cell that contains our coding exons and our non-coding introns. And this gets transcribed into messenger RNA, which just includes our exons, the coding bit, and that goes on to make our protein. And here's my plumber analogy from last year. So if you have the blueprint of a house, it is sort of similar to DNA, so you have all the information included to make our house. A plumber will come along, and he doesn't need all of the information in the house for the house. He only really cares about the plumbing in the bathroom, let's say. So he's going to take some scissors to these blueprints, and he's just going to cut out the information that he actually wants to make the plumbing work. And now that he can make sense of what he's actually got, he can go to the site and actually make the protein. So pre-mRNA splicing that I was talking about before is sort of this process here where we go about editing our information to actually make it make more sense to the cell. So here are the exons again that I referred to as the coding bits, so the actual bits that contain the information. And we have our non-coding intron here. And we see that there are important sequences within that intron that allow the cell to recognize that it's a bit of non-coding information that needs to be removed. So this is done by these proteins that are in the cell. And they will bind to these certain specific sequences and allow these two exons to come together and the intron to be removed. So mutations that disrupt how these proteins bind can really mess up how this message is made, and the resulting protein might not be made properly or as needed. So my PhD is focusing all about like what variants within that non-coding region are really important for our cells and what is causing disease. But today I wanted to talk about the antisense oligos. I'm just going to shorten oligonucleotides to oligos. That they also bind to these particular sites, which means that our proteins that are trying to splice this thing can't see it because there's essentially a roadblock in the way. So these cellular machineries will look elsewhere to bind to the intron because of these oligos that are just in our way. So going back to our Emd gene, I've made up this sentence here, or copied it from Google, and to sort of show how these exons of coding information stitch together. So when correctly spliced, we read, "The big mad cat ate the fat rat and the red bat and was fed." So if we were to splice incorrectly and we splice to the middle of this exon here rather than at the beginning, we'll lose this little bit of information about our rat. So the next sentence will read, "Big mad cat ate the rat and the red bat." So we're losing a bit of information, but we can still read to the very end of our sentence, right? The general message is still there. However, if we were to completely skip that exon and this bit of information here, what actually happens is that we have a frame shift issue. So something I forgot to mention earlier is that this sentence specifically reads in threes. Each word in this sentence is a three-letter word because our code is read by our cells in a triplet sort of formation. So if you delete anything not in a series of threes, you really shift and scramble up the message as shown down here. So it'll automatically align everything in threes. So now our sentence doesn't align because we've deleted 5 base pairs. And so we get absolute nonsense, and we can no longer make Emd because our message has been completely scrambled. So can we control this sort of splicing to our advantage? So Duchenne's muscular dystrophy, as some of you may know, most common dystrophy affecting 1 in 5,000 males, early onset, and it presents in a progressive limb girdle pattern of weakness. So back to our Wikipedia sentence of eteplirsen triggers excision of exon 51 during pre-mRNA splicing. I'm zooming up here. This red box is just the region where exon 51 is, so that coding bit of information to make the protein dystrophin later on. And that's this middle one here. And I've applied the same sentence as an example. So when this splices together correctly, we see the sentence reads as it should. However, when we use the drug eteplirsen, it'll bind to exon 51 as such. And then when we splice it, we see that, we've messed up our triplet reading frame. We've got 4 letters here, so we're going to have to rearrange that, and now we've scrambled our message. Like, hang on, this is a drug that's supposed to have therapeutic approaches. Why are we scrambling this? This doesn't make any sense. So let's think of an example where we have a patient who has Duchenne's muscular dystrophy, and it's because they have exon 50 missing. So now they never had exon 50. They had this massive deletion in their gene, so that when they try to splice their exons together, they also have a scrambled code. So if we give this patient the drug eteplirsen, then they will also ignore exon 51. And yes, we're missing this information, and we're already missing this bit of information, but our sentence actually reads correctly. Like, we can actually get to the end of our sentence now, even though we are missing a little bit in the middle. Like, the cat just seems to have eaten less today, I suppose. Yeah. So you might be thinking, like, well, surely if we have the information to make the protein there, why is it okay just to get rid of it? Surely this information does something useful. So what dystrophin actually does is it sort of connects these muscle contractile proteins in the cell to membrane proteins. So it just really holds everything together in your muscle cell, being like this sort of structural thing. And I like to sort of think of these muscle membrane proteins as like a boat. And dystrophin is the chain that sort of holds the anchor and sort of holds everything together. So if we're missing a few links of the chain in the very middle, then that's actually okay. We just have a slightly shorter chain, but we still have that same structural integrity that we need for our muscle cells. If we, however, were to mess up either end closer to the anchor or the boat, then we might run into more issues. But because the deletion is in the middle of the dystrophin protein, we can mess around with it a bit without having terrible consequences. So now I'll talk about the clinical trial that was released last year by Sarepta, who produced the drug eteplirsen. So they recruited 12 boys who were eligible, so ages between 7 to 13, who had a variant where by using eteplirsen, you could put the instruction back into frame and unscramble the code. So examples that they used, so I used the example of exon 50 deletion, but there are a few other exons around there that if were also deleted, eteplirsen would then put it back into frame, and we could get to the end of the instructions and still have a normal protein. And they also had to be able to walk 180 to 440 meters on the 6-minute walk test. So they were treated for 180 weeks, and I think they're still being treated at the moment. Like, this is an ongoing trial. They'll have weekly doses of eteplirsen, and at the end of the 180 weeks, 11 of them had biopsies so that we could assess the levels of dystrophin that were in their muscle at the end. And they performed the 6-minute walk test again. So what they found was that there was a slower decline in performance of the 6-minute walk test because it is a progressive disorder. They were just progressing less with their disease as the untreated controls, which was fantastic. Also a relatively stable respiratory function compared to controls. And what they found when looking at the actual muscle tissue itself and seeing, you can actually look at the instructions and what instructions are being made. And yes, we did see that exon 51 was definitely being skipped in these samples, which is good, the drug is working. And 9 out of 11 had significantly increased levels of dystrophin in their muscle. And so one way they look at assessing this is they use a technique called immunohistochemistry. And here's an example of what it looks like. So these are just from our own lab because I couldn't find any controls in the paper. But each one of these circles represents a muscle cell. And the red is dystrophin that has been stained with this fluorescent protein. And so you sort of excite it with a laser in a dark room and you see this red right? Like red, dystrophin, great, good. And and we can see that all the red is at the membrane of the cell, because that's where I said it was doing the most structural linking to try and keep the cell together. With patients who don't have any dystrophin, we see that it's just completely black. We don't see any red at all, because there's just no dystrophin being made, because we can't get to the end of the instructions because we've scrambled somewhere in the middle. It might be hard to see on the screen actually, but there are a bit of patchy redness that you might be able to see. Might be a bit too dark, particularly in L here. But anyway, so A and B, there wasn't much red, and you can see little flashes of red. So there was improvement. Like, it's not 100%, but there have been studies to show that you don't need like huge amounts of dystrophin to live a normal life. It's good news that the drug is penetrating into the muscle and actually making proper dystrophin again. The problems with eteplirsen at the moment is that it only works for 13% of patients with Duchenne's muscular dystrophy. So you have an already rare disorder, and within that subset of is another level of rareness that your mutation affecting dystrophin has to be fixed by one specific mechanism of skipping exon 51. It's very expensive, about $300,000 US a year, not currently available in Australia. Because clinical trials are small. As you saw, they only had 12 patients because it is a really rare disorder and a rare set of mutations. So how many patients do you need to sort of show that it is a beneficial and a cost-effective drug sort of thing? It's difficult. But this does open doors for other potential therapies using this technology. So it could theoretically be applied to any sort of genetic disease, so long as manipulating that splicing process and maybe skipping over the mutation, or maybe playing with that using this technology could work. And there are at least 28 clinical trials that are currently planned and are in progress using this technology, which is exciting. And we noticed within our own cohort of patients with neuromuscular disorders that we have at least 5 people who by making one of these little oligos would put the reading frame back into play and would allow them to make a fully functional protein. And my supervisor, Professor Sander Cooper, is really keen to go down this track and trying to create a therapeutics team that manipulates splicing to help people with neuromuscular disorders. So the challenge with this is recruiting enough people who have the same variant and the same rare disorder. And there's been lots of talk about maybe fast-tracking clinical trials with drugs such as these so that we can get them to patients sooner, particularly because this is such a personalised medicine. Like, these drugs will be so specifically tailored to people that it's not really it's not useful to do a clinical trial, but a really rigorous one. So they're working with that in the States at the moment with the FDA to see if we can fast-track these. So I just want to say thank you, Muscular Dystrophy New South Wales, for providing me with my scholarship. It's really helped out a lot. And everybody in my team, and as well as people we collaborate overseas. Last week, actually, we managed to publish my first paper in the American Journal of Human Genetics. So this paper sort of looked into mechanisms, a new mechanism of a splicing defect. So looking at the intron, going, okay, what's this? And found that there was one particular type of variant that was causing these issues. And with this, we were able to diagnose 2 of our patients. And one family, and this is their quote, meant that they were eligible to a certain drug that really helped them become ambulant again, which is fantastic for them. Thank you. Oh, Antisense. Yes, I didn't explain it. So if you remember at the beginning of my slides, if I can go back to the beginning. Yeah, sorry. Here. So basically, you have each one likes to pair to another strand, so you have the 2 strands pairing. So when we say antisense, we basically make a oligo that is the opposite of this strand. So yeah, what I should have mentioned is that an oligo is, so if that's your DNA, your RNA, because we've gone from our DNA, transcribed it into RNA, and that's in a single-stranded state, which means it's got all this lovely base pairing that could easily happen like that, right? So an antisense is sort of the opposite pairing mechanism. So this would be the antisense strand of that DNA. So basically how it works is your antisense oligomer is a synthetic strand of base pairs like this, and it will just bind to your RNA. So it just has the specific opposite sequence or antisense sequence to bind to where we want it to block it. Yeah, that makes sense.
Any more questions?
Oh, there's one here.
Sorry, I'm hogging. $30,000 a pop a year, 300,000 a pop a year. Wonderful technology, but unreachable for just about anybody. I mean, how does this change?
Yeah, from my understanding, I think that's just because this particular drug's they've been developing it for over 25 years, and I think by the time you have so much IP involved, and that's sort of the cost. But I think, and please, anybody feel free to correct me if I'm wrong, but I think the actual technology to actually make it itself is cheaper. It's more the IP at this point that people are paying for. But hopefully in the future it'll become more accessible.
Hopefully.
Yeah.
Hi, I just want to know, how did you target your subjects for the study?
Yeah, so we haven't. So in this particular study, they just made sure, just looking at the sort of variants in play, would be like, okay, if we were to delete exon 51 in this particular scenario, would it turn it back into frame, sort of thing? So on the Wikipedia page, they talk about exon 51 amenable by exon 51 skipping. So it takes a clinician and scientists to sort of look at the sequence and sort of decide whether or not it's this drug would put it back into frame and give us the product that we want or not.
And how people approach you guys to be targeted? I mean, how's that working in terms of organizations or promotion, or is muscular dystrophy in charge of this, or how's that?
For the stuff that we do, because we haven't actually set up any therapeutics yet, we're still sort of thinking about it, but what happens at the moment for what we do is clinicians will find an interesting variant in a patient, and they'll go, I'm not sure if this is going to affect splicing or not. Oh, I know Sandra Cooper's team, they know all about splicing. I'll ask for their opinion. They send us an email. What do you think about this variant? And we say, oh, yep, great, send us some samples over here. We'll have a look. We'll try to confirm if it's disrupting this process or not. And then the clinician can then take that information and help the patients. Yeah.
And the short answer is you talk to your doctor,
Yes.
and hopefully they will be aware of what.
Yeah.
Great. So on that note, we really thank Sam for talking to us and congratulate her for her paper.
Yeah.